Now we're gonna move on to antibiotics that target protein synthesis. And this is something that I want to mention. We never mentioned that in the second unit. Could, but ribosomes in bacteria. Well, we kind of mentioned it really. Ribosomes in bacteria and ribosomes in eukaryotes are different, which means we can develop drugs that would target ribosomes in bacteria, but not in eukaryotes. Are you following my lead? Which means if we can inhibit a ribosome in bacteria, these drugs are going to have high specific toxicity as well. Does that make sense? So, let's talk about the protein synthesis inhibitors. So here, we target protein synthesis. Okay? So our first target will be 30S subunit of the ribosome. Now, there are two classes of antibiotics that target 30S subunit. First class is aminoglycosides, which distort the ribosome, and it results in the production of faulty proteins. Okay, and the second class,
what do they distort?
the ribosome itself. So the structure of the 30S subunit gets tweaked, disturbed. Does that make sense? And it results in misreading the codons. So wrong amino acids get inserted. Does that make sense? And second group is tetracyclines. Which inhibit the tRNA attachment. So they basically stop synthesis of proteins from happening. Does that make sense? Good? Okay, awesome. Again, those are classes. Here, we have inhibitors of 50S subunit, the big, large subunit of the ribosome. Okay, first one, macrolides, streptogramins, glucosamines. It's kind of my test for Alzheimer's that I still remember those. Okay? Now, what these guys do? They inhibit the polypeptide chain release. I'm going to expand on this. First of all, let's take a little pause, okay? Aminoglycosides, neomycin, kanamycin, gentamicin, actinomycin, not used. Kanamycin is used kind of frequently, others are not so common. Does that make sense? Tetracyclines, you're probably familiar. One of the best broad spectrum antibiotics we have. Doxycycline is really popular to treat anaerobic gram-negatives and intracellular infections. We good? Macrolides, Z-Pak. Streptogramins, streptogramin, glucosamines, clindamycin, for instance. Okay, now what does that mean, polypeptide chain release inhibition? When we did our representation of protein synthesis, remember that? The polypeptide chain was kind of sticking out of the ribosome. These antibiotics don't let it to get out of the ribosome. See what I'm saying? And that it gets stuck, so no more synthesis. Another drug that belongs to that same same group right here, 50S subunit inhibitors, is chloramphenical. An interesting drug that prevents the addition of new amino acids. Essentially, bless you, it inhibits the enzyme called peptidyltransferase. Okay? Does that make sense so far? We good? And finally, I put it kind of separate. The group of drugs called oxazolidinones, they inhibit the formation of initiation complex. So why we put this drug kind of in a separate category? Look, these drugs inhibit the ongoing process of protein synthesis. Does that make sense? You're good? This drug prevents synthesis from happening. Initiation complex doesn't even form. Any questions so far? My very ambitious goal is to squeeze all the drugs into that thing, so you will have the final picture. Okay, we're good? Moving on.